Hello, my friends. Thank you so much for joining me. Today, I am going to do a presentation on the manufacture of monoclonal antibodies and using the quality by design approaches. This is the second recording because the first one didn't work. So many apologies if you've downloaded the first one. You might have heard me speaking halfway through, I disappeared. So anyway, this presentation, we're going to be looking at the design aspects on the monoclonal antibody and on the cell substrate itself in order to improve yields and in order to have a more efficacious clinical performance. So this presentation, as I said, is an overview of the development of the monoclonal antibody and how we can engineer them to maximize clinical performance and to minimize adverse drug reactions and also to optimize manufacture because you can make certain changes or adaptations to the protein sequence in order to bring about better manufacturing process as well as better clinical performance. I'm also going to give an overview of the manufacturing process for a humanized monoclonal antibody in the context of quality by design. So the first is design of molecule, design of the molecule and the quality attributes. So where do we begin? How do we design a molecule? In this case, this biological entity, which is a humanized monoclonal antibody. What quality attributes should it possess? Well, the first thing that we do and the first thing that we take into consideration is the disease, the disorder, the condition that we're tending to, we're looking to treat. We start off with a target product profile. This takes those aspects into consideration, the clinical aspects. It takes into consideration the efficacy, the safety, what exactly do we want to see happen, what is our proposed mechanism of action in terms of how the molecule is going to interact with the target protein, what is the mode of action, so what is the outcome, what is the clinical effect that we want to see. All of those scientific assessments on efficacy and on safety, what potential side effects do we expect to see? Like, for example, issues with site, the administration of the site of administration might present adverse drug reactions in terms of how the drug is administered. That's what we expect to see. What other safety concerns or adverse drug reactions can we expect to see? The other thing we need to take into consideration are the drug product aspects. So, for example, compatibility. When we're administering the drug, what are we going to be dissolving the drug or mixing the drug with? What are the agents, the dissolving agents, the saline perhaps, or the dextrose solution? What is the compatibility of the drug with those? What's the shelf life? In-use shelf life. What is the shelf life of the actual drug product itself? Why would we want a drug product? We would want to engineer a drug product that could have a reasonable shelf life in order to make it commercially viable and in order to make it a practicable drug. And how is this drug product going to be presented? What is it going to look like? So those are the sorts of things that we start off with when we're considering the design of our medicinal product. We may consider it from a drug product target protein development program, or we can consider a phenotype or phenotypical development program where, yes, we may be targeting a protein, but we're looking mainly to alleviate symptoms as opposed to targeting the actual disease. So there are various different development programs and formats that can be used, and the quality attributes are the properties of the molecule or the characteristics of the molecule that serve for that intended use. What I really find interesting with regards to this presentation, or what I found interesting, was the fact that a lot of the experience that we've gained with regards to adverse drug reactions, with regards to potential issues with manufacture, we have refined the engineered antibody that can overcome some of the problems that we've experienced in the past. So that's what I refer to here, the design features, in terms of not only the mechanisms of actions, but the design features that are designed within the molecule to minimize adverse drug reactions, to maximize clinical potential. And we use the knowledge that we've gained from prior knowledge with regards to cell substrates, and this is the CHO-derived information. Knowledge that we have about these isotypes, the IgG1, knowledge that we have with regards to the mechanism of actions. So what type of therapeutic mechanism do we want to see? Do we want to see just the binding effect and the, if you like, neutralization of the particular protein or the particular target? Or do we also want to see not just that, but we want to see a an immune response, so the effector response, the ADCC and the CDC response? And this is a humanized antibody. What are the advantages of that, and what do we want with regards to dosing? So all of these things are part and parcel of the design of the molecule, and all of this is incorporated in the quality by design approach. So there's a slight mistake in this, but I wonder if you can spot it. But basically, antibodies consist of a heavy chain and a light chain. Here I've described the heavy chain features. I'll show in another slide the light chain features, and I've also got the disulfide linkages that keep the structural and the conformation of the antibody, and that's really important for its ability to bind with the target protein or the target epitopes on the antigen. So let's just start from the beginning, I guess, and that is that antibodies are part of the body's adaptive immune system, and they're produced by these B cells, and they're, as I said, highly selective. They're specific to their targets, and their targets are what we refer to as the epitope, and the epitope is on the antigen, and the antigens can be on viruses, it can be on cancer cells, it can be on cytokines, or cytokine receptors. The one that I gave in the previous presentation, this was used for, rheumatoid arthritis and Crohn's disease, and basically the cytokine was tumor necrosis factor, and the antibody bounds with that particular cytokine and therefore reduces its effect. So that tumor necrosis factor is implicated in inflammation pathways for this rheumatoid arthritis, and the antibody binds with, it's an antibody fragment, it binds with the tumor necrosis factor, therefore reducing that inflammation and having that therapeutic effect. So it's kind of phenotypical in its approach, in that it doesn't necessarily target the underlying cause of the disorder or the disease, but it targets the outcomes, which are the symptoms, which are inflammation. So that's kind of how antibodies work, but they can be used diagnostically as well. They can be used for immunological assays, for ELISAs. They can be used to quantify or track a target of interest. So they're really versatile and useful, products and components. It's not necessarily clear here, but the antibody has several key regions that carry out their functions, and I will go through them in another slide. So here I'm showing the light chain, the variable regions of the light chain and the constant regions of the light chain. The constant regions are really responsible for this FC effector functions. So they are on the heavy chain as well as the light chain. I don't know whether they're, so I certainly know that the FC are responsible for the ADCC and the CDC. So ADCC is the immune response. It's the antibody dependent cellular cytotoxicity, where you basically kill the cell of interest through apoptosis. And complement dependent cytotoxicity is where the complement system is activated, and sometimes these antibodies are known to activate the complement system. Sometimes they are not, they don't. So this is the light chain, and light chain consists of a variable region and a constant region. And basically the variable region in the heavy and the light chain, they contain this complementary determining regions, and there are 3 regions on the heavy chain and there's 3 on the light chain, and that's a total of 6. And these CDRs, complementary determining regions, they're responsible for having the paratopes, and the paratopes are what interacts with the epitope on the antigen, and they bind together. So the one of the important regions, the variable regions, are it's highlighted on the light chain, but I've also given you the previous slide where it's on the heavy chain, and that's where you get the binding, or at least you get the complementary determining regions that possess the paratopes. So you can genetically engineer, or we can now genetically engineer, different forms of antibodies. So we can engineer the fragment, which is just looking at the heavy and light chain variable region of that antibody, and just simply that. And so we're looking not to generate any FC functions. We're just simply looking at binding and having the therapeutic effect that way. We could also be looking at chimeric antibodies. So this is where we graph a variable region onto a second antibody. That could be a human antibody. And that's where you think about having a humanized form. And this is to reduce the adverse drug reactions, because obviously you don't want to be, you don't want to be administering an antibody that's going to have an immune response, and it will do if it's non-human. And then you have antibody fusion proteins, where we fuse the heavy chains or the single chain for the variable region onto an antibody. You can also have combine antibodies to have maybe 2, where you have bispecific antibodies, or 3, that's trispecific, or multispecific, where you have multiple antibodies that are bound together using protein engineering techniques. So these are some of the forms, not all of them, for genetic engineering. And you can produce these antibodies for various different therapeutic applications. Right, so this particular structure that I'm referring to is an IgG1 monoclonal antibody, and I just want to talk about how the structure imparts and impacts upon the efficacy and safety. So the quality by design approach that I talked about earlier could be used to build attributes and characteristics into the antibody to maximize clinical performance. Again, I said that previously. So what do we know? We understand that glycosylation patterns and the glycosylation profile is important. It's important for its mechanism of action, and it's important for its therapeutic efficacy. As I said, if you have glycosylation profiles or patterns that are different to human, that could potentially lead to the body recognizing the monoclonal antibody as non-self, and then instigating an immune response, which could be an adverse drug reaction, and that would would or could lead to the removal of the monoclonal antibody from the body and the body system, which is of no value whatsoever. Since glycosylation is, a post-translational modification, it's less well controlled compared to, say, for example, the genetic sequence and genetic expression of the sequence. So transcription and translation is better controlled than this glycosylation post-translational. But we can develop a means of engineering the host cells to to control those glycan sites where glycosylation occurs, so that we get what we want. So the correct required glycosylation in order to get the therapeutic effects that we're looking for. So, for example, I'll talk about this in another slide, but you can introduce various mutations into the sequence that will potentially modify the antibody, or you can also engineer the cell substrate in order to do this. I put those disulfide links here and I've identified them. They are known to shift. Disulfide links are there to maintain the structure of the antibody and thus to maintain its stability and its efficacy. When you get shifts or breakages in your disulfide links, then you're likely to get degradation, or you're likely to get changes in the molecule's activity, where it can dimerize or form different aggregates, and obviously that will impact on its ability to bind. In fact, it will probably change completely and not be efficacious. So if you can introduce residues, amino acid residues that stabilize these disulfide links and mean that you have a more stable product that is less prone to aggregation or dimerization, then that is something that can be engineered, and you can design that into your molecule. Again, deamidation is another potential problem with antibodies, and you want to prevent deamidation most certainly at the antigen binding site, but also at the FC site. So you have this FC, the fragment that is constant or crystallizable, and then you have the Fab, which is the fragment with the antigen binding. And you want to, well, it really depends on how your antibody works. As I said, if you have this FC region, and if that FC region is responsible for the effector function, which is this antibody dependent cellular cytotoxicity that you actually want, you can enhance it, but you also don't want to get deamidation along those regions, the FC region or the Fab region, and so you can engineer your antibody to produce residues that prevent that. So antibodies can be designed to increase its half-life in vivo by engineering it and building that quality and making it improving its clinical performance. I did talk about disulfide shift, and I did say how each light chain and heavy chain contains a disulfide bond within the variable domain as well as the constant domain, and they help to stabilize the structure, and they're responsible for ensuring that or maintaining the correct folding of these antigen binding sites. So therefore you get your specificity, you get your affinity for your target antigen. So if those bonds break, you can then reduce your antigen binding ability and disrupt the whole whole mechanism that the antibody adopts. So your variable region, your Fab bonds, your antigen binding bonds, your disulfide bonds are critical for your antigen binding stability, and the constant region, that's the FC bonds, the disulfide bonds are essential for the antibody structure itself, the effector function, and that's the ADCC or CDC, the complement dependent cytotoxicity, and the half-life. Yeah, and as I said, one of the problems with disulfide shifts is aggregation or degradation. Disulfide bond shifts can weaken your antigen binding. It can impair your immune system activation if it's the FC part of the antibody. It can promote aggregation and reduce the antibody stability. All of these things, they do compromise the therapeutic or immune functions of your antibody. So these disulfide bonds, they play a critical role in maintaining the overall stability of your antibody. And as I said, shifts could lead to fragmentation, or it could lead to aggregation or degradation, and it could reduce the half-life and the effectiveness of your therapy. So I want to talk about secretion. So antibodies are usually secreted from your cells, from these cell substrates, and they're secreted into your cell, your media. And because these molecules are secreted, the actual antibody itself can be engineered to contain appropriate signal peptides to the light chain and the heavy chain, and this can facilitate secretion, and that can work towards optimizing a good yield of your antibody, and therefore, the peptides that you select, they can be optimized they're attached to the heavy chain and the light chain, and they can be optimized to determine which ones are the best ones for secretion in your cellular system. And this is really good because obviously you want to get good yields. You want to have a good enough that's the problem with biological biotech products, is the yield. So you want to do anything you can do to optimize the yields, is something that you definitely would be aiming for. The other thing here I've got in this diagram is glycan sites. So glycan sites is where you get your glycosylation, and antibodies, which are of course glycoproteins, these sugars are attached to these glycan sites, and these sugars modulate the ADCC functionality of the antibody. So basically, antibodies are produced naturally, naturally by B cells, and they are produced to modify. You don't want antibodies being overly active, and then you have problems, autoimmune diseases and disorders. So that's the purpose of these sugars, to modulate the ADCC functionality. However, if you're designing a drug where you want to enhance that ADCC effector response, you can then design a drug where the glycosylation patterns are modified to get the best outcome. So you can obviously those glycosylation patterns have to be human-like in their profile, because if they're not, then that can potentially lead to the body recognizing the monoclonal antibody as a non-self, and then instigating an immune response, and then that could be an adverse drug reaction, or lead to the removal of the monoclonal antibody from your body, and then of course that is an adverse drug reaction because you're not getting an efficacious outcome. But this post-translational modification of glycosylation can be engineered to produce, you can engineer the cell substrates, and you can also engineer the transgene to produce the amino acid residues that promote, for want of a better word, the required glycosylation that you need. So these disulfide shifts, linkages, they're very important. Again, as I said, they're important for the structure, and we can also engineer the monoclonal antibody with amino acid residues that prevent the breakdown of these disulfide bonds, or the shifting of these disulfide these disulfide linkages, and that's again how we can build quality into the molecule itself to ensure you have a stable product. Yeah, again, the FC chain is important for the structural integrity of the antibody, as well as the effector functions. Specifically, the FC chain is responsible for. So we want to, as much as possible, if that is an important part of your mechanism of action, or a key or critical part of it, then you want to avoid or remove any particular protein residues that can cause aggregations, and so you can design your molecule to achieve that. And this is useful for stability, and it's useful for manufacture. As I said, I don't know what I did say, deamidation is a problem as well that we are often faced with, and again, this can be along the FC or Fab region, can be problematic, and can, in the Fab region, it can cause issues with binding. In the FC region, it can cause issues with regards to the effector function. So it's something that we are aware of, and we need to, if possible, engineer into our an antibody or into the cells themselves to prevent. So I just wanted to give this example of the fucose residue and how the removal of this fucose can help with enhancing the ADCC effector response. So the ADCC effector response is a natural thing that occurs, but this fucose residue mediates that response. So removing the fucose from the antibodies can be achieved by genetically engineering the cell lines that produce them. So there's the CHO cell lines, or the HEC293 cell lines that produce these antibodies that contain these fucose. If you engineer those cells by knocking out the genes that are involved in fucose biosynthesis, or the attachment of the fucose to the glycans, then you get this afucosylated antibody, and this has an enhanced ADCC activity. Yeah, so all of these attributes, they are all determined inside of the production bioreactor, which we'll talk about later. So the FC receptor on cells, they bind to special proteins on certain immune cells, such as natural killer cells or macrophages. So when the antibody binds to the target, the FC receptor, well, the FC receptor binds to the natural killer cells, I believe, and that triggers the natural killer cells to become activated, and this is ADCC. That's my understanding. I could be wrong there, but anyway, in this particular case, what you're looking at is this ADCC. This ADCC effector function, that's what you're looking at, and you want to enhance it, and so you have this FUT8 knockout or double knockout where both copies of the FUT8 gene are deleted, and this FUT8 is responsible for either adding the glycan, the fucose to the glycan structure. And so what you want to do is you want to remove that so that you can enhance the ADCC effector response. So the strength of the interaction between the sugar glyconstruction, and the antibody is dependent upon that fructose. So if the antibody has fructose in the glycan, then, or in its glycan, then it's weakly bound to the receptors. But if the antibody is afucosylated, it binds much more tightly, and it has a stronger immune response, and it's better at destroying target cells. I just wanted to just quickly put this slide in, and this is relating to transfection of mammalian cells utilizing plasmids, because I'm not sure what everybody's background is, but I just realized that it might not be clear how that transfection occurs, because there are various ways of transfecting genetic material into cells. You can do it via the viral route, or you can do it via the plasmid route. And the plasmid route has its advantages and disadvantages, but if you are using this route, it will be the pi-mediated transient transfection, and this is basically where this molecule pi, it forms or a complex, if you like. It forms a complex with the DNA, and this is what facilitates its entry via endocytosis into the mammalian cells and then into the nucleus. Right, this next part is about the manufacturing process, and I'm just going to give a top level overview. So the manufacturing process consists of two sort of stages, I tend to think of it, or two sort of phases. You have your upstream manufacture process and you have your downstream manufacture process. Your upstream manufacture process is all the stuff that's going on from the cell expansion right through to the production of the antibodies and harvesting. Your downstream manufacture is where you're clarifying your bulk. It's where you are using various techniques, to usually chromatographic techniques, but and chromatographic and filtration techniques, to purify and to concentrate and to ensure that you have the correct buffer in your system. And then you have your quality control. Now, your quality control comes last, depending on the type of manufacturing process that you have. If you have a traditional batch manufacturing process, then your quality control testing will be at the end, after you've done your manufactured product, and it is more for confirmation. Your quality control can also include your in-process control testing as well, as that you conduct during your manufacturing process. So that's included as well. But where we talk about batch manufacturing, we're specifically referring to the confirmatory aspects of your monitoring process, where you're just confirming the quality, but you're not actually testing quality into your material. The quality is built into your manufacturing process. Just another note here on your culture conditions, because they're very important. Your cultured conditions can determine the quality of your cell substrates when you're during cell expansion, and they can also determine the quality of your antibodies once you have manufactured those antibodies and they're ready for use. So you want to have as an optimal cell culture conditions as possible. The cell culture conditions have to be relevant to the operational step that you're doing. So, for example, your expression vector media conditions are going to be product dependent and specific. So are your transient transfection going to be specific. But once your transient transfection occurs before you clone select your cells, the cells that you're going to use for your cell substrate. So even though it's important to have this tied down, even though it's important to have this noted, it's not part of your routine manufacturing process. Your routine manufacturing process is where you take your cells from your cell bank, your working cell bank, and you expand those cells. And your culture media for that, and your culture duration, all of those things have to be determined, has to be characterized, and has to be used to boost your yields and reduce degradation. So cell culture media is important because the quality of your host cells is impacted by your cell culture media, and subsequently the quality of the antibody is impacted by your culture media. So let's talk about the seed culture. So this is basically the process that we're using to generate the cells, the CHO cells, the CHO cells that are responsible for, or the factory houses that produce the antibodies. So we start off with maybe one or maybe more than several vials of your working cell bank, and you expand the cells in initially disposable vessels. These disposable vessels will contain the cell culture that has been optimized for the best conditions and the conditions that are going to yield optimal expansion, and you want to remember that the cells, the quality of the cells that are produced are directly related to the quality of the antibodies that these cells are going to produce. So you start off the seed culture, which is the expansion of the cells from the working cell bank in these disposable vessels, and these disposable vessels are usually sort of plastic bags, or they're flasks, and they're single-use plastic containers, and they're used in the early growth stage, and they're convenient because they reduce the risk of contamination and they don't require cleaning after use. And these cells are expanded step by step in an increase in larger disposable containers until they reach the sufficient quantity that you can transfer them to a stainless steel bioreactor. So once you've transferred them to this bioreactor, this is the fixed stirred tank bioreactor. They're made of stainless steel. They are not disposable. They cleaned and reused for multiple batches, so you have that potential for cross-contamination or of adventitious agents such as microbial adventitious agents, but also cellular adventitious agents, although you would want to reduce that, but it depends. In simple terms, like you start off with a tiny batch in a small disposable vessel, you then expand your cells, get a large enough concentration of cells, viable cells, that you can move it to a larger third tank bioreactor once you've grown enough. And then you keep on expanding until you have enough for your full-scale production. Your large production bioreactor, this is where you ultimately want to produce your antibody. So this is where the cell growth occurs, your cell growth where you want to get that initial viable cell concentration before you go to your antibody production, in which you would then change your conditions in order to induce that production of the antibodies. The WV stands for your working volume. So your bioreactor could be large and it can have a working volume of 15,000 litres. So it will probably be larger than that 15,000 litres. And in this large production bioreactor, the fully expanded cell or seed culture is transferred for this final production. They continue to grow until they reach their peak density, and this is the initial viable cell concentration, and you want to have that concentration tied down because you want to have a reproducible manufacturing process that reproducibly and consistently gives a certain yield. So you want to, as much as possible, tie down and control certain aspects. And then eventually they will then actively produce the desired monoclonal antibody when you provide the correct nutrient feed. And as I said, it has to have the controlled environment for that antibody production. So that might be different to the environment and nutrients that are going to be required for your cell expansion. The production bioreactor also has sensors and automated control, and this is to control the manufacturing process in situ, in place. So since this is where the production of the antibodies occur, the quality attributes of the monoclonal antibody is determined in this particular environment, and therefore it's really important to have controlled conditions and your specified growth material, or your growth media, or your optimized growth media. So, as I said, and this is just a summary slide, the purpose of the bioreactor is to produce the high quality monoclonal antibody production. You have to start off with your initial viable cell concentration, and then you have the correct conditions and the correct specified cell culture media in order to produce enough monoclonal antibodies to be harvested from your culture media once your production peaks. So once the cells in the large production bioreactor have reached their peak density and they've produced enough monoclonal antibody for that particular period, the next step is to harvest your product. And this involves usually just centrifugation and depth filtration. Centrifugation is where you remove the cells themselves and you're left with the supernatant. So the cells are sort of like the crud at the bottom of the vial, the centrifuge vial, and it's just a process of separating out the cells from the antibodies that are in the supernatant. And then this is the crude, if you like, and then you put it through depth filtration, which can further remove larger host cell proteins and host cell materials, or any other material that shouldn't be there. And then you go through these purification stages. So the first step that I've got here is protein capture. I mean, you may very well do some more things before you go on to your chromatographic steps, but this is just what I've got to summarize. So your protein capture is basically using affinity chromatography, and basically that is exploiting the differences between your component, which is your antibody, and the other materials or the other components in your media. Antibodies, this FC region, the constant region, specifically binds to a protein A, or selectively binds to this protein A, and that can be exploited in affinity chromatography, allowing the antibody to be captured on the protein and all the other things that are not wanted to be washed away. And then you change the mobile phase, or the eluent, to a specific buffer, low pH, high salt concentration, that disrupts that binding between the protein A and your antibody, enabling you to capture the pure antibody, or at least the free antibodies, in a purer state. And then you do your polishing steps, which will be ion exchange chromatography, which could be anionic or cationic exchange, and they basically separate proteins based on size exclusion chromatography. Anion exchange or ion exchange chromatography separates your protein based on charge, not on size. Size exclusion chromatography separates your components based on size. So you get aggregates, you might get dimerized antibodies. These are different, have different molecular sizes, molecular conformations, and they can be separated by size exclusion chromatography, further purifying your manufactured material. And then ultrafiltration. So ultrafiltration is a membrane-based separation technique, and it just uses the size exclusion principles, if you like, to retain the larger molecules like the monoclonal antibody, and it allows the smaller molecules like water and salt to just pass through it. And it's just a way of you can use it to concentrate the antibody solution by removing all the excess water and small solutes. And then diafiltration is another process that's similar to ultrafiltration, and that's useful to exchange your buffer. So you want to, you want to have a formulated buffer with a desired formulated buffer that will improve the stability of your antibodies and the efficacy. And this is what diafiltration can be used for. Ultrafiltration and diafiltration can be performed in continuous manufacturing, but it can definitely be performed in batch manufacturing mode, where fresh buffer is added while the filtrate is removed, ensuring your efficient exchange. Yeah, and basically you just want to ensure you get your final formulation. You might need to adjust your pH. You might need to add stabilizers. It would be specific to your product. You might perform an additional sterile filtration to remove any bacteria, so that would be 0.2 micron filters, or you might perform an additional viral filtration. I don't think that you'd perform that at the later stage. And you'll then do your filling and your filling of your vials, or filling into bulk containers, and before freezing or presentation in its final dosage form. For your quality control testing, now, as I said, you can perform quality control during your manufacture. That's not the same as your in-process control testing, which is different. But quality control testing, in my mind, is routine testing that's usually performed after the batch of drug product has been manufactured. You perform it to predefined specifications, and it's really confirmatory. But process analytical technologies is different, and that can be incorporated for continuous manufacturing, where you get real-time release testing, and that's a much more agile and efficient way of manufacturing your product, particularly when you're looking to scale up or produce more of your antibodies, and scaling up and scaling out might not necessarily be the route that you would go for. So this is just an overview flow diagram of the downstream process, where you get your clarified bulk, clarified through centrifugation and depth filtration, and then you use chromatographic techniques to further purify it. So this is protein A, but you can use other proteins dependent upon the antibody. You subject it to low pH incubation to remove any viruses, any enveloped viruses, and then you cation and exchange chromatography and anion exchange chromatography to further purify it. You may then do this nanofiltration for small virus retention, and then you might do your formulation using ultrafiltration and diafiltration. You may then fill your bulk in bulk containers, or you may decide to freeze those bulk containers, or you may decide to fill them directly into the vials. So here is an overview of the process, and I have a slight error there. So it's protein A affinity chromatography. So you can use different types of proteins. You can use another protein called protein G, but protein A is what's usually used in this case study. And it's just there to capture the monoclonal antibody, as I stated, and to allow all the other process impurities to be removed. And those could be host cell proteins, it could be DNA, it could be small molecules. You then link these onto ion exchange chromatography techniques to further purify. Your low pH incubation is where you perform your viral inactivation. So these are specific to enveloped viruses, and this is to reduce the potential for adventitious agents that may be or could be present in mammalian cell cultures. Chinese hamster ovary cells are a well-established cell line, and if you have good GMP procedures and good training procedures with regards to the professional or the personnel that's carrying out the manufacture, then in theory you would want to reduce the potential for adventitious agents, but you can't. There are some things that you can't control, so you need to have in your manufacturing process actions that mitigate the potential for introduction of adventitious agents. In any case, this is specific to enveloped viruses, and later on we will see the nanofiltration for non-enveloped viruses. And then you have cation exchange chromatography, which is another polishing step to remove aggregates, to remove host cell proteins. Basically, chromatography, what it does is it exploits the difference between the antibodies or the resins or the stationary phase from the products and the eluent that's being used to deliver them. So it's just a method of separating and purifying. Anion exchange, again, is separation based on charge. You remove host cell proteins, DNA, protein A, and endotoxins, and virus as well. And then you would perform filtration that I discussed earlier, and this is for the removal of non-enveloped viruses. Ultrafiltration, diafiltration, as I've explained, is a way to obtain the correct formulation, to further filter, further purify, and to ensure that your drug product is in the correct buffer for the formulation. And then your final fill-in in your bulk containers or fill-in in your vials for presentation. So I'm just going to talk about the challenges related to manufacturing processes for biologics in general, but it can apply to monoclonal antibody. What you want to do is you want to build quality into your manufacturing process rather than end testing. And this is important, I think, if you want to have a design space. If you want to have a design space that can include various scales, and if you want to start that with that strategy at the start and throughout your initial development, because there are potential challenges relating to scaling up or scaling out with regards to biological or biotechnological products. Scaling out has an impact on your cost of goods, and it's applicable for most modalities. You start off with a smaller scale and then you want to increase your scale as your market expands and increases. But particularly for biotechnological products, it can present challenges, and the reason being is that the manufacturing process is the product itself. So if you make changes to your manufacturing process, potentially you're making changes to your product. It can be viewed that way by some health authorities. So scale has a very important impact on the cost of goods. It's particularly so for biotechnological products, but it is challenging for scale up and scale out. Even in countries where comparabilities studies that start off from initially from an analytical perspective, you may be able to show that analytically there's no change in the quality of your product, but the regulators may demand that you perform additional animal studies, non-clinical studies, and additional clinical studies. So that can be more time consuming, and there can be an additional expense just because you want to scale out or just because you want to market the product in another country. So, you know, you really do need to maybe perhaps consider looking at scale-up effects during the definition of your design space, to perhaps potentially, you know, reduce the friction, if you like. It doesn't mean it's going to necessarily be the case, but reduce the friction or the challenges that regulators and health authorities may consider. Your manufacturing steps in the process have to be manufactured to GMP in all stages, and at all stages for your biological biotech products. And this is due to the biological nature of the products. You need to be able to, in some cases, trace the cells and reagents, particularly if those cells were used from human origins. And there are other legislations that might apply. In the European Union, it's the EU tissue directives, and you may have to show traceability for 30 years. So there's additional legislation and regulatory perspectives that have to be taken into consideration anyway when you're choosing to market a biotech or biological manufactured material in another region. And in addition to that, scaling up and scaling out may be considered to be a real challenge. Even for chemical entities, when you want to change your batch size, increase your batch size significantly, scaling up, in some countries, I know for example, Brazil, that is considered to be like a new marketing authorization application. So, and that's for chemical entities. So it's not really, I don't know, it's not the same as in the EU. The EU maybe perhaps take a different approach, but I know that for chemical entities, if you want to have a scale up, if you don't include that application during the marketing authorization application, say if you've got your product marketed in another country and you want to increase the batch size, if you're going through a marketing authorization application, you can include that additional data in, say, for example, a response to question, so that you have it all assessed at the same time. But if you get your marketing authorization approved certainly in Brazil, and you want to implement a batch size increase, then you could be looking at you would be looking at another marketing authorization application. And I know it's the case also for some other countries as well. So you have to really consider looking at scale up effects and scale out effects during your development, but ultimately there may be more regional requirements, and that may involve more cost and more time. I also want to talk about analytical testing and this real-time release testing, which is a fantastic approach, particularly for continuous manufacturing. It could significantly reduce those lead times. But even with real-time release testing, you still need to perform microbial testing, sterility testing, which can be quite time-consuming. Although I am aware of the fact that they have these shorter tests that can be used. I want to talk about continuous manufacturing and the concept of perfusion in the next slide. So, perfusion, I guess, is a form of continuous manufacturing in biologics. Continuous manufacturing can consist of one stage or one unit operation that can be continuously manufactured, and all of the others may or may not. It doesn't have to be from start to finish. So it can be part of the manufacturing process, can be a continuous manufacturing. And in this particular case, the cell culture used in biological manufacturing in the production vessel can be adapted, if you like, for continuous manufacturing. So it basically involves a constant flow of fresh culture media into your bioreactor while simultaneously removing waste and spent media. And this allows the cells to be viable and productive for extended periods. And so basically what you have is a steady state operation. You've got your cells continually producing the desired product over extended periods of time. And in theory, you get a consistent product quality. You've got stable conditions and you've got reduced variability. And in theory, you have a greater yield and higher productivity because your cells are retained for longer and they are viable for longer. You do need to qualify this, and you need to look at the stability of your cells and justify this from a scientific perspective as well as from a risk perspective. But it reduces your downtime as well, so you don't need to do so much setting up of the process or cleaning between batches. So it's effective in terms of your operational management. And you can integrate some downstream processing. So for example, you can look at continuous chromatography or chromatographic techniques, but you have to also take into consideration the challenges with regards to the lifetime of the column itself, because columns ultimately have a certain lifetime, and columns have to be equilibrated before they can actually be used for their required function. So there's all sorts of things you have to take into consideration. It's not sort of continuous and it just goes on forever. But well, nothing is, but there are those considerations to take into consideration. So you can integrate it with downstream processing, but there's a caveat with regards to some of the steps in your downstream processing. Perfusion is increasingly being adopted by biopharmaceutical manufacturing as a means of efficient, scalable and cost-effective production of your drug product. So yeah, this is what I was talking about earlier about scale effects and incorporating those in your design space, if that's possible. If it's not possible, and if you haven't done that, then you would need to do the required additional scientific and maybe even clinical and non-clinical data generation in order to justify your scale up. Process analytical testing, this is where you test during your manufacturing process, and then you better control and adjust, and you know, it's a better way of monitoring rather than depending upon confirmation at the end, although we do in-process control testing. The use of inline sensors for monitoring and direct control. This is usually for viable cell concentration and glucose. The potential to use spectroscopic techniques such as HPLC, I think that's already been established if you check the guidelines. And again, rapid microbiology safety tests to reduce the time taken for release. So currently, I guess the traditional format would be to do your sterility test, and that can be quite time consuming. So thank you so much for listening to my presentation. I've gone over what I'd previously planned, but I hope that I've covered some of the points that I missed out in my previous presentation. If you've got any questions, please don't hesitate to answer, and please don't hesitate to leave a like if you want, or a comment. Thank you so much. Bye bye.